Good day, this is Professor Will, CBMD PhD, and today is Monday, June 29, 2020. It is 6:26 and 33 seconds a.m. Eastern Standard Time, and this is a continuation of my corona research. But before we get started, let's take a look at something I wrote. In today's video diary, I would like to highlight the fact that I've done incredibly well since spring and especially since the beginning of summertime. My routine has been fantastic. I raised the bar on my nutrition, walking, restricting my social interactions, and being explicit and able to cope correspondingly. Moreover, inconsistency to my research and planning these fabulous videographies which make an intellectual impact. I create these academic notes to help me recite and reinterpret as an exercise towards my intellectual pursuits. Furthermore, as you take a glimpse at today's videography, you will spot my attentive and extensive interest in bioinformatic investigations. Life is a mystery and full of spontaneous surprises and discoveries, accompanied by destiny and a world of entropy. I consider myself a discoverer, a streaming Renaissance polymath. My wintertime goal is to design my own bioinformatics 3D geometric interface in addition to a database which will take tedious amounts of time management, another delicate operation. Thank you for listening to my videography, which I believe are purely educational experiments. So let's hop over to my corona research. Proteins are active because of their spatial conformation and the occurrence of active centers, which interact with other molecules. Quantitative studies of these features can be accomplished in various ways. A direct approach involves computation of protein folding based on energy functions. Again, dynamic programming can be used to reduce the computational burden. If this is accomplished, or if the structure is known from X-ray crystallography, it is possible to consider computations of active centers of proteins based on the geometry of their surfaces. The interaction of proteins may be approached computationally by solving the docking problem, employing methods of computational geometry similar to those used in robotics. These and related computations are involved in time and memory consuming. An alternative approach is based on the notion of evolution as a laboratory. By following the evolution of biomolecules, it is possible to infer their function and the relationships between them. An example of this approach is the evolutionary trace method of Lichtarge. In this method, homologous amino acid sequences from a number of species are used to infer a phylogeny. Subsequently, this phylogeny forms a basis for classification of the amino acids in the sequence, based on their conservation in the branches of the tree of different order. The amino acids which are conserved best are likely to belong to the active center. This method has led to the confirmed predictions of the active sites. Similarly, the Fallenstein-Churchill algorithm mentioned above allows identification of amino acids which have evolved slowly. These will be candidates for belonging to the active center. The new branches of bioinformatics will require the creation of new databases and continued work on purely informatic structures such as ontologies which allow retrieval of information with a very rich structure. At present, virtually all branches of science use mathematical methods as parts of their research tool. Science has entered a phase of mathematics invading other disciplines. This is because the concepts in all areas of science are becoming more and more mature and precise, and mathematical tools are flexible and generalizable. Without exaggeration, we can say that the majority of the methods of applied mathematics are used as tools in bioinformatics. So, is there anything peculiar about using mathematical modeling in bioinformatics? Among the tools of applied mathematics, some are of special importance, namely probability theory and statistics and algorithms in computer science. A large amount of research in bioinformatics uses and combines methods from these two areas. Computer science algorithms form the technical background for bioinformatics in the sense that the operation and maintenance of bioinformatic databases require the most up-to-date algorithmic tools. Probability and statistics, besides being a tool for research, also provides a language for formulating results in bioinformatics. Other mathematical and computational tools such as optimization techniques with dynamic programming, discrete mathematics algorithms, and pattern analysis methods are also of basic importance in ordering bioinformatic data and in modeling biological mechanisms at various levels. What is mathematical modeling? By mathematical modeling, we understand describing and reflecting reality by using formalized tools. Models can be of very different types: stochastic or deterministic, descriptive or mechanistic, dynamic or static. Mathematical models can pertain to phenomena in many different areas, for instance physics, chemistry, biology, engineering, or economics. How do we develop models? Models are developed by combining, comparing, or verifying hypothesis versus empirical observations. We develop models by using the laws of nature, physics, chemistry, and biology. We apply principles of conservation and/or variation extremum principles, which lead to balances in differential or difference equations for the evolution of the state of a system. Models can include discrete events and random phenomena. What is the benefit of using mathematical models? Using mathematical models allows us to achieve a better understanding and to organize better our knowledge about the underlying mechanisms and phenomena. Sometimes models can change qualitative understanding to quantitative knowledge. Models can allow us to predict future events from present observations. Models can be helpful in programming and planning our control and design action. What is specific in modeling in biology and molecular biology? Compared with models in physics and classical chemistry, models in molecular biology pertain to more complex phenomena. Following from this, there is usually a greater extent of simplification that needs to be applied when building the model. A large individual variation leads to a substantial element of randomness, which needs to be incorporated into the model. An element which is present in all models is simplifying hypothesis. The benefit in using a mathematical model is often related to solving the compromise between the extent of simplification and the model and precision in predicting data. Complicated models are usually less reliable and less comprehensive. Oversimplified models can ignore important phenomena. Research work that forms part of modeling involves model building or model learning, applying the model to the data, and model modification. After enough experience has been gained by repeated application of these elements of modeling research, models often start bringing benefits. One crucial element is verifying a model versus the data, which very often starts from fitting free parameters of the model. This involves tasks such as identification and parameter estimation solved by various methods of static, dynamic, and stochastic optimization. Among optimization methods, the least squares method deserves special attention owing to its reliability and very vast range of application. If one assumes model with many free parameters, one has substantial flexibility in fitting the model to the data. Extreme cases called black box modeling, which means fitting the parameters of standardized models to the measurements without inquiring about the nature of the underlying processes and phenomena. Both fitting to data and analyzing the predictions of mathematical models is done by using computers with appropriate software. Some very useful programming environments are very high-level programming languages for supporting engineering and scientific computations. MATLAB, Mathematica, Maple are some several computational examples. For some specialized tasks, one may need programming languages of lower level, such as C, C++, Delphi, and Java. Facts in biology and biochemistry become established when they are seen in a biological or biochemical experiment, or better, in several independent experiments. Knowledge develops in biology and biochemistry in this way. There are two aspects to the development of biology and biochemistry concerning its relation to bioinformatics. First, with the development of experimental techniques, the number of findings and discoveries in biology and biochemistry has become so large that efficient access to the information requires the use of the most advanced informatic tools. Second, browsing and analyzing data in bioinformatic databases allow or helps us to predict facts in biology and biochemistry or to propose new hypotheses. These hypotheses can be used for designing new experiments. There are several well-established paths in which bioinformatics can be applied in this second way. After the genome of a new organism has been sequenced, then by using knowledge about the structure and organization of genomes and the contents of genomic databases, researchers can find the genes and compare them with their homologs in other organisms. Inside genes, coding sequences can be identified leading to amino acid sequences of proteins. These approaches can be used in a variety of types of research. Information obtained from comparing genomes can be used for inferring the ancestry of organisms and also for predicting the functions of genes and proteins. Comparing sequences of amino acids and proteins in different organisms allows one to infer their functionality of important sites and active sites. By combining computational methods with browsing protein databases, one can improve the methods for drug design. For example, when the sequence of a virus causing a disease has been found, then it is often searched for regions coding for proteins. Next, using the hypothesis that these proteins are important in the activity of the virus in the human organism, design of the appropriate treatment can focus on drugs blocking their activity. Bioinformatic databases contain massive amounts of experimental data. Browsing and analyzing these data is fascinating and will surely lead to many interesting discoveries. Developing projects concerned with searching for interesting information in bioinformatic databases belong to the most vital area in scientific research. It is important to stress here the interdisciplinary aspects of the research in bioinformatics. Search through bioinformatic databases is often initiated by posing a question related to some biological problem. The bioinformatic project then involves designing the computational and algorithmic aspects of the search or browsing. The results are most valuable when they lead to answering the question, to improve understanding, or to interesting biological interpretations. Probability calculus involves the computing of probabilities of random or stochastic events, defining probability distributions, and performing manipulations using random variables. Statistics is concerned with presentation analysis and inference based on data obtained from experiments with an element of randomness. In a random setting, events may happen or not, and defining the conditions of experiments involves assigning probabilities to events. The probability of an event is a measure of the frequency of its occurrence under repeated controlled conditions. Large collections of probability distributions associated with typical physical or biological experiments with a random component, as well as the rules of probability. Calculus are presented in a large number of references. When fitting probabilistic models to data, it is commonly necessary to estimate parameters of distributions of random variables. We cover methods of parameter estimation, with emphasis on minimum likelihood. We also develop some more practical computational aspects, including the EM algorithm, with examples of its applications. We include some topics on testing statistical hypotheses. On that note, we're going to jump over to dimensionality reduction principal component analysis. A need for dimensionality reduction arises when the number of features is large. Experimental results in molecular biology and biochemistry often lead to the creation of a large number of measurement data points. Examples are gene expression intensities in DNA microarrays, proteomic spectra, and data concerning conformations of large molecules such as proteins. In such situations, the number of features, measurements obtained in each experiment, is much bigger than the number of experiments. One expects that only some of the measurements will be correlated with the state of experiment under study. Two cases are possible. The first possibility is that the state of experiment, e.g. deceased versus healthy, is known. A related problem is to select the subset of features most suitable for differentiating between experimental states. The second possibility is that inference must be done solely on the basis of the set of feature vectors, without any knowledge about the underlying structure. Good day, this is Professor Will, CBMD PhD, on infectious disease, molecular microbiology, and pathophysiology. Well, hope you enjoyed listening. Until next time, have a great day.